At DISPENDIX, we are committed to partnering with biotechnology companies to improve the efficiency of synthetic biology workflows. Using the I.DOT Liquid Handler, researchers can miniaturize and accelerate DNA assembly and make NGS library prep less costly and more efficient for QC of gene synthesis. The I.DOT dispenses from any well to any well, enabling the automation of complex assays. Faster, more accurate, and volume-verified transfers will revolutionize any workflow. Rapid pooling of oligos or DNA fragments from library plates in under 1 second can save 10 to 15 hours for high-throughput labs. Reduction of volumes down into the nanoliter range dramatically cuts reagent costs, saving precious samples and preventing dilution errors. With support from the application team at DISPENDIX, we can have your synthetic biology workflow up and running from day one at a precision and speed that will surprise even the most experienced automation engineer.